Hello, everyone, and welcome to today's broadcast, Bacteria That Eat Electricity, Understanding Biocathodes Using Culture-Independent Techniques. It is presented by Brian Eddy, PhD, a research biologist at the Center for Biomolecular Science and Engineering at the Naval Research Laboratory. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located in the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Eddy. I will now turn the presentation over to him.
Hi, and thank you for giving me a chance to talk to you today about bacteria that eat electricity and about the ways that we're trying to understand how they're able to do it. So to start off with, electron transfer is life. So all living things use electron transfer reactions in order to generate electricity or generate energy and to generate biomass. So everything that we're familiar with, or the animals and things like that we're familiar with, like elephants and butyric acid bacteria, use reduced electron donors like carbohydrates in order to provide electrons, which then they transfer to an electron acceptor. In this diagram here, oxygen. In other organisms, they'll use other things such as nitrate or sulfate or iron oxides, or in some cases, organic molecules as the electron acceptors. And then there are other organisms like trees and any photosynthetic organisms that are able to use electrons from water and then energy from light in order to reduce carbon dioxide and make more biomass and make more organic matter. And the reason that we're interested in this today is because there are some bacteria that are able to use extracellular electron donors or electron acceptors. So in our case, we use oxygen as an electron acceptor, and that's something that we breathe in and it gets inside of our cells, and that's an internal electron acceptor. But there are bacteria that are able to actually transfer the electrons from inside their cell to the outside, to an electrode that's called an anode that acts as an electron sink. And then there are other organisms that are able to use what is called a cathode in order to accept electrons from that cathode. And then they're basically pulling electrons out of the cathode. And what we're interested in the Navy, in this case, is how these organisms are able to do that and how we might be able to use that in order to look at things like biocorrosion or microbial fuel cells. And we've got a keen interest in using synthetic biology in order to try and make better organisms to do these things for us. And it comes down to, in the beginning, the Navy was mostly interested in microbial fuel cells from a power generation perspective, because these are things that can be used as long-term stable power sources that would be able to supply energy for instruments and things like that. So the pictures that I show on the screen here this is a buoy on the right that was holding some instruments that was being powered by a microbial fuel cell that was buried in the sediment. And this lasted for around a year until a hurricane came along and wiped it out. But it was the first proof of principle that you can power a sensor array for a long period of time using just bacteria and whatever they're able to find to eat. And from this, then we've also started looking at how organisms are able to use the cathode in order to live. So we actually took, and an electrode from one of those microbial fuel cells and then some additional seawater and some sediment, and used it to inoculate a culture that then has been transferred dozens of times since then. It was initially brought into the lab in 2009 and has been maintained since then. And this is a bacterial community that's been growing on here that is very robust. It's been shipped around the country to other collaborators to work with, and it's transferable. We can just keep scraping it off and putting it onto a new electrode and it will grow. And what's really interesting about it is that one of the organisms in there is autotrophic. So it's able to use the electrons from the cathode to fix carbon dioxide, and then it's feeding the rest of the bacteria that are there. The key thing here is that only a few of the bacteria that are growing here are actually things that we've been able to cultivate in a pure culture in the lab. And one of the ones that we haven't been able to grow is the autotroph. So we've been using some culture-independent methods to try and understand how it's able to do that. And our goals are to try and understand extracellular electron transfer from a very fundamental level. So previously, a lot of work has gone into the engineering aspect of things. So what can we do to make the electrodes different so that bacteria will be able to grow better using them? And what we're interested in is specifically how the bacteria are using their proteins in order to grow, so that we can try and improve things from that side of things. And like I said before, one of the things we really are interested in is synthetic biology. And what we're trying to do here is find some chassis organisms that we can put components into, and then find components to put into them that are going to be more environmentally relevant, that will actually grow in seawater. To date, most synthetic biology has been done in E. coli, which does not grow very well in seawater. And so we're trying to make things that would be more relevant for the Navy. And like I said, we don't have cultivated isolates of some of these organisms. And that presents some challenges compared to how most microbiology has been done in the past. So what we're trying to do is understand how a community is growing when we can't bring the individual parts into the lab separate. We have to study them all together. And so what we're trying to do is go from that slimy muck on an electrode to nice clean models of what electrons are doing in these organisms. And to kind of step back and look at things from a very high perspective here. The way we're doing this is we're looking at part of what's called the central dogma of biology. So for everything that's alive, there are these four things that are the genome, which is made up of DNA, the transcriptome, which is made up of RNA, the proteome, which is made up of protein, and then the biochemistry that is occurring in life. And some of these things, like the genome and the transcriptome, we can actually measure very precisely with a very high throughput and with relatively few biases, although there are still some biases there. However, things like the proteome are a lot harder to measure to really get a good idea of what's going on with every protein in the organism. And biochemistry, it's really not possible at all to study that at a very high throughput way. So we're focusing on the metagenome, or the genome and the transcriptome of these organisms. And the way that we're doing this is with metaomics. So this is basically looking at all of the genomes in a community and all of the transcriptomes in a community at the same time. And what we're learning from the metagenomics is what genes are in this community and what things this community might be able to do. So we can look at genes that are known from other organisms and say, these organisms that we see in the community may be doing ammonia oxidation, or they may be doing nitrate respiration. Then we can look at the metatranscriptomics and see which of these organisms that were there in the metagenome are actually active. And then we can see which genes are active and how do they change expression depending on what the growth conditions are. We've also got people, other people in the center, not me, doing proteomics and metaproteomics and metametabolomics, trying to look at communities using these techniques. And they're very important, but they're a lot harder to do, and there are there's a lot of things that get missed when you look at it that way. And then we're also working with isolates, trying to pull out individual organisms, because that's where you can end up getting the most fine resolution of your data. And then obviously you need to have an isolate in order to do a lot of the manipulations that we want to do. And so to start off with, we're going to look at metagenomics. And I know there are other metagenomics talks in this meeting, so I'm not going to go into too much detail here. But basically, the way this works is you have a community, you sample its DNA, and then you do sequencing, which gives you millions and millions of short reads, which by themselves aren't very useful. But then you can take them and you can assemble them into larger contiguous sequences, or contigs, and in some cases even an entire genome. And then that gives you a better idea of which organisms are there, their relative abundance, which ones are very abundant, which ones are very rare, and in some cases you can get some other information from that. And so what we're trying to do here is, again, just look at which genes are present in this community and what might this community be able to do. And so with our data, we started off with kind of a first pass looking at a single sample, which makes it harder to pull out individual genomes. But we were still able to group these sequences after we'd assembled them into what looked like 15 to 20 different organism dens. And in this community, we were able to identify almost 80,000 genes total, which is a whole lot of data. And we're trying to sort through that, and then trying to look at which components we were interested in. But then we also had more data that was coming in, and then we ended up rebinning everything with 8 of our biological replicates. And then this really gave us some very good bins. So we had 16 bins of genomes that were almost completely complete. And one of these in particular was really interesting. So this is the one that's in red here, that was initially identified as an unclassified Chromatiaceae. And that is it's a group of bacteria. They're known as a purple sulfur bacteria, although a lot of them aren't purple and a lot of them don't actually use sulfur. But there are some representatives in there that are autotrophic. So that immediately was a key finding. So we were really interested in it. And when we looked in the rest of the metagenome, we see that this is really the only organism that looks like it's an autotroph. But then it also has some genes that are also found in iron oxidizers, which we hypothesized would be an analog of the kind of metabolism we are looking at, because those are bacteria that are able to take iron that's outside the cell and then take electrons off of it and then grow using the energy they can get from that. And so we wanted to look at this organism in a little bit more detail. And then I should also say that about the time that we finally got these good bins out of this, we submitted some DNA to PacBio, because they were interested in assembling metagenomes from using sequencing from their sequencers. And they got back to us a couple of weeks later with a complete genome from this organism that we had spent the last year trying to assemble. But using that, we could be very confident that we did do a good job of assembling, and that all of the genes that we thought were in there are actually in there. And then using those genes, we could go through and look to see where it fits in the tree of life. And one of the interesting things that we found from this is that if you just look at the 16S ribosomal RNA gene, or 23S also, you get a very different picture of what kind of organism it is than if you look at the whole genome itself. And so if you just look at the 16S, it looks like it's a member of the Chromatiales, which I had talked about before is the purple sulfur bacteria. However, if you look at the whole genome, it looks like it's more closely allied with the Thiotrichales, which are a different group of bacteria that is also, some of them are autotrophic, but they're not photosynthetic. And recently, just a couple of weeks ago, there was another paper that came out using the whole genome technique that has reclassified them as a complete separate order. They're still within the Gammaproteobacteria, but they are now separate from both the Thiotrichales and the Chromatiales. So this is a really interesting new organism that so far no one had found. And then actually in the last few years, more and more people have been finding it now that they're able to put this 16S gene that they're finding in the environment with this organism. and be able to tell that it's not a member of the Chromaceales, it's something else entirely. And so, one of the other things that we'd wanted to look at is just where in the biocathode biofilm this thing is growing. So to do that, we use fluorescence in situ hybridization, which is also called FISH, to specifically label each of these organisms based upon their 16S or 23S ribosomal RNA sequence. And from this, we can see there's a clump in this slide that you can see that is labeled as Candidatus Tenderia electrophaga, which is the autotrophic organism. And the probe itself is green, but when it's mixed with the blue from the DAPI, which is the DNA stain that stains everything, and the red, which stains Gammaproteobacteria, which this organism also is, those all combine to make this kind of orangish, whitish color, which is very confusing because then the alpha proteobacteria, which are a completely different group of bacteria, are also orange. But if you look at the individual colors with this microscope, you can pull out that this clump that's labeled here is actually Tenderia, while the others are not. So it forms a patchy biofilm. In this case, it's not a very dense one. And other electrodes that I've looked at, it does get more dense. But one of the things that's really interesting about the fact that it's so patchy is that if we look at electrical conductivity, we see a very highly conductive biofilm. So this is work that was done by my colleague Matt Yates, and what he's doing is using what's called an interdigitated electrode array, or an IDA, to look at the conductivity from one electrode to another. So on the left side of the slide, you see a diagram of what these IDAs are. They're basically two comb-shaped electrodes that are mixed together, kind of like interdigitated. They're like stick your fingers between each other, but there's a very defined gap of 5 micrometers between these two electrodes, and these form a sinuous path that goes back and forth. That's always 5 micrometers apart, and the whole path is 13 millimeters long, which gives you a very long gap, basically. And then he's measuring the conductivity of electricity between those two electrodes across this 5 micrometer gap so he's measuring the different potentials by holding so there's a source and a drain electrode, and he holds the one at a slightly higher potential so that electrons will flow from the source to the drain. And then as he changes the median potential of those two electrodes, going from high to low and then back, you can actually see that at very low potentials, there's no conductivity, but as the potential increases, then you get conductivity at a peak of around 550 millivolts. And if you look at the total conductivity, it's averaged over what we think is the size of the biofilm on here. You actually see that it's as conductive as a geobacter biofilm. And these are some bacteria that have been studied for a long time. They're thought to make one of the most highly conductive biofilms that's out there. So it's very interesting that this patchy biofilm is as conductive as that. And we're still working on trying to do this in a way to get a little bit more information about that. And then another thing using these IDAs, one of the interesting things we can do here is only poise one of the bands. So these organisms can only grow if we give them electrons at a certain potential. And if we only set one of these electrode arrays at that potential, then they're only able to grow on one of them. So you can see on the left, there are arrows that are pointing to bands that were poised. And then on the right are bands that were not poised. So the Tenderia is only able to grow on the ones where we're actually feeding them electrons at the right potential. And as the biofilm grows, eventually it spreads out enough that it makes a connection with the unpoised bands, and then those actually they're poised at the right potential also for things to grow on them, and then you can start to see a little bit of growth there. Looking back at this organism Tenderia, this is the one organism in the biofilm that's actually able to fix carbon dioxide. And then it also has genes from iron oxidizing bacteria that make it look like it can perform extracellular electron transfer. And so if we look at the CO2 fixation pathway, this is using the Calvin-Benson-Basham cycle, which is actually the same cycle that plants use and cyanobacteria use. But it's also found in a lot of other bacteria. And what's particularly interesting about this organism is that it has two of these rubiscos. So it has one, the form 1AQ, which is normally thought to be adapted for a high carbon dioxide and low oxygen environment. And then another, which is the 1AC rubisco, which is adapted for a high oxygen, low carbon dioxide environment. And so the 1AQ is somewhat less efficient than the 1AC, but the 1AC has a much higher overhead. So it requires a lot more proteins because it actually forms these carboxysome shells, which concentrate the rubisco and concentrate carbon dioxide in the same spot so that it can be more efficient. And that'll come in a little bit later. And then the other thing to point out here is that the reductant for this CO2 fixation pathway is NADPH. And we'll talk a little bit more about thermodynamics in just a minute. But while I'm on the subject here, I also wanted to say that we have these proteins in here that are able to do, that we think would be able to do extracellular electron transfer, or EET. So we've got some that look like the Psych2, which is on the figure on the left, the far left complex there is the cytochrome C2 or Psych2. And then we also have some multi-heme cytochromes with a porin that's located near them in the genome, which look like it may be able to do things similar to what you see in the figure on the right. So these are the ones that we think are the most likely to be involved in getting electrons inside the cell, but we have yet to actually prove that. So to get into the very basics of the thermodynamics here, one of the things that this organism has to be able to do in order to grow is make the reducing equivalents, or the NADH or NADPH, in order to fix CO2. And the challenge here is that we're giving them electrons at 310 millivolts, and they somehow need to do this reduction reaction that is at minus 320 millivolts. So somehow they've got to get energy to drive this reaction in reverse. And the way they do that is likely to be reverse electron transfer. So what happens then is we have electrons coming in at 310 millivolts and then going through the forward direction to cytochrome CBB oxidase, which is generating a proton gradient that then is used in order to drive the reaction in reverse. And we'll talk a little bit more about this later, but I wanted to introduce that now because it's going to be kind of key to understanding some of the data later. So right now I'm going to start talking more about the metatranscriptomics of this community. And so what we're interested in from this is basically what organisms are active and which genes are being used. And this gives us an idea of what this community is trying to do. And we're going to look at two different conditions to see how what it's trying to do changes depending on what the condition is. Why we're using metatranscriptomics is, if we go back to the central dogma of life, there are different control points within this chain where you can stop or start the next step in order to control how things are going to happen. And the easiest place to start and stop something is right at the beginning. So we stop it right after we basically stop the formation of the mRNA that would be then used to make proteins. And then this lets us look at which genes are being used, because we can look at how much of each specific RNA is present within the cell. And then we can look at how this, the level or the amount of RNA for each gene changes depending on what growth conditions we're using. And the way that we're doing this is we're using the metagenome that we have, and we're aligning the RNA-seq data to that in order to see which genes are being used. And I know this is a very complex figure, but the main thing to take away from this is that as we take the data from the metagenome, we take the data from the metatranscriptome, and then we're combining them, and we're looking at the activity of the individual members of the community, and then looking at the activity of the different genes in order to get an idea of what's happening. And to do this, we're using this kind of paired electrode design. So we are growing this, we're growing 4 of these electrodes up, and then we're measuring cyclic voltammetry to make sure that everything electrochemically looks normal. And then we're switching 2 of those electrodes from the optimal potential, which is 310 millivolts versus a standard hydrogen electrode, or in this case, in the figure that's on the right here, I've got it against a silver chloride electrode, where it's 0.1. This is all just, it's relative to a reference electrode. So I normally just use the standard hydrogen electrode, but so we change one of them, or we leave one set of reactors at 310 millivolts, and then we change 2 of them to 470 millivolts, which is a much less favorable potential for them to grow, because they get less energy from the oxygen reduction reaction, which is at 810 millivolts, and it takes them more energy in order to get the reducing equivalents they need for CO2 fixation. So it makes it a lot harder for them to grow when we put them at the higher potential. And then we harvest the electrodes for RNA and then sequence it. And then we actually repeated this again a couple of weeks later to get 4 more reactors. So we have basically 2 reactors at each of 2 potentials at each of 2 inocula, so 2 by 2 by 2. And so then if we look at which organisms have the most RNA total in these reactors, we can actually see that Tenderia is the most active organism that's in our reactors. And this is really interesting because if we just look at the DNA, it only makes up around 1/4 of the DNA. But then if we look at the messenger RNA, it's around 3/4 of the messenger RNA. And the other organisms, some of them, Marinobacter and Labrinzia, which here are in green and blue, are more abundant based upon the DNA, but then less abundant based upon the RNA. So this is particularly interesting because we think that Tenderia is the organism that's feeding everybody else. So it has to be the most active in order for everything else to be able to grow. And then we can also look at how the percentage of activity in Tenderia is correlated with the amount of current being generated. So even though this is a relatively reproducible bacterial community, there's still a lot of variability. So our reactors will range from around 10 millivolts to around 50 millivolts, or milliamps, sorry, 10 milliamps to 50 milliamps, depending on just how things shake out and how the community develops. But if we look at the percentage of Tenderia within that community, we actually see that there is this correlation between a community that has more Tenderia and a community that's making more electricity. So this was another thing that's supporting the idea that Tenderia is the organism that's actually transferring the electricity, or transferring the electrons from the electrode. If we look at some of the other bacteria that are in the community, we actually see a negative or no correlation with the amount of current that's generated. And so even though this is only based on 4 reactors for each potential, if we look at the electrodes or the reactors that we did for the metagenome also, if we look at the DNA, the relative abundance of the DNA within the community, we also see this same trend where the reactors that are making more current are also more enriched for Tenderia. So, total that ends up being something like 16 reactors. So that's a pretty good number of replicates. If we focus in still on Tenderia here, we're looking at the individual protein complexes and groups of genes that perform a specific type of metabolism. And one of the things that came out of this is that if we look at the CO2 fixation genes in particular, one of the things we see is when there's more energy available, so when we have them at the optimal potential, then we see a higher relative abundance of the genes for CO2 fixation. And then we also see this form 1AQ rubisco, which is the one that's less efficient but faster. And then a few of the other blue, I guess, gene modules that we see here are things for glycogen formation, which is basically a storage product, the pentose phosphate cycle, which is part of the central carbon metabolism. So this is one of the bits of metabolism that basically makes all the things that make up a cell. If we look at the genes that are more highly expressed at the suboptimal potential, we see the genes for this form 1AC rubisco, which is the more efficient but higher overhead one, and then also the carboxysome this fits in there. So this is suggesting that at this potential they're actually trying to be more efficient, even though it's going to be a less fast way to grow. So we can also go through and look at individual, I guess, chunks of the electron transport chain and look at how they change their expression depending on what potential the community is grown at. And so we see a few groups here where at the top we have genes that are more highly expressed at the higher potential or the suboptimal potential. And then down at the bottom, we have some genes that are more highly expressed at the optimal potential. And we can kind of form some hypotheses based upon which genes these are and how they're used. So, you know, the high potential genes, a lot of these, so we have the Psych2, which is going to be involved in getting electrons into the cell. And then we have some of these genes that are going to be in the forward electron transport chain. And then down here in the middle, I have what's labeled as the RET chain, or the reverse electron transfer chain. And these are the genes that are going to be involved in basically generating and reducing equivalents. And one of the things we see here is that they don't really change their expression very much at all. So it seems kind of like this is being held stable, while the things on the right side of that electron transport chain are what's actually being varied. And so what kind of comes out of this is that there are actually multiple routes for electron transfer within this organism and within this. So this diagram here is also based upon a lot of work that people have been doing with iron oxidizing bacteria and looking at genes that are shared between iron oxidizing bacteria and Tenderia in order to get an idea of which ones might be used for this. And then we also looked at how they change expression depending on the potential here. And what we see is it looks like we have basically on the right side in the box that's labeled terminal oxidase, we see this set of genes here that's labeled CCO-plastocyanin. This is one of the terminal oxidases that would actually be reducing oxygen and generating a proton motive or proton gradient that then would be used for energy by the cell. And so here I'm just going to kind of summarize what we've talked about so far, and then we're going to go on to talk a little bit more about some of the energetics within this community. So far, what we found is that Tenderia looks to be the keystone organism. So it's autotrophic, it's changing its activity depending on the potential, it's changing the expression of genes depending on what potential we're growing it at, and it's very abundant. One of the things we don't really know yet also at this point is what role some of the other organisms that are in this community are going to be playing. That might be a good question for later, is to, what these other things are doing. So what we're going to look at now is how these potential routes for electron transport might be used, and we're going to look at how we might be able to tease some of this apart. And so the way we're going to be doing this is with these specific metabolic inhibitors. So these will block a particular part of each of these chains, and from that we can kind of start to pull out what these things are doing. And so this is a very old idea. People have been using these inhibitors for 40 or 50 years in order to block a particular thing. And they've been used in order to provide some of the first evidence for the reverse electron transfer, or in this case, it's called uphill electron transfer. And that's basically what we're trying to do here. And so what we're looking at is the electron flux through each of these potential routes. So we've got, on this slide, I'm showing 4 different inhibitors. Sorry. So on this slide, I'm showing 4 different inhibitors, and each of these is going to block a specific part. And then using the Nernst equation, we can actually kind of predict how much electron flow we're going to see through both the forward and reverse. This equation basically lets us look at the potential of electrons, of the electron transfer reaction from the cathode to the cell itself, and then the electron transfer from the terminal oxidase to oxygen, which is going to be at around 816 millivolts versus standard hydrogen. And then also from the cathode to NADH, which is around minus 320. And then from that, we can actually predict how much electron flux we need to have through each direction of the pathway based upon the metabolism we know these cells are doing, which is the CO2 fixation reaction. And from that, we can calculate that they'll need around 75% of the electrons to go through the forward pathway, and around a quarter at most to be going through the reverse electron transfer pathway. And so the first thing we're going to do here is basically test that hypothesis using this inhibitor called CCCP, which collapses the proton gradient. So in order to drive reverse electron transfer, you need to have protons in the periplasm. And what the CCCP does is it basically grabs onto those protons, and then it can carry them across the membrane and collapse the gradient. So we think that when we do this, what we're going to do is basically stop all reverse electron transfer, but we think there may be some increase then from the cytochrome C oxidase that may actually counterbalance a little bit of that. And when we do this, we actually see that we get this immediate drop in current. If you look at the graph on the left, I guess I should point out here also that when I refer to drop, I mean a drop in the absolute value, the magnitude of the current. So because this is a cathode, it's pulling electrons off. We are recording the current as being negative just because of, it's kind of the arbitrary way that we look at electron transfer. So it looks like it's an increase in current when it's actually a drop in the absolute magnitude of the current. So when I add the CCCP, what we get is this immediate drop that ends up being around 25% of the total current. And then we get this much slower increase or much slower approach to zero in the current, which we're interpreting as the cells attaching, because in some cases the attachment of cells is actually ATP dependent, so they need energy in order to stay attached to the electrode. And the other possibility is that they're just deciding to leave because, if they can't breathe on the electrode anymore, then they may as well go somewhere else and try and find a better place to live. But either way, we see this immediate drop in current. And what's particularly interesting about this is that this is also correlated with the amount of current that they were generating to begin with. So what we think is happening here is that under under conditions where they're basically happier cells, they're able to generate more energy per electron, and thus when we add the CCCP, we get a bigger drop in current. So some of this may be related to the amount of oxygen that's able to get to the electrode itself, or local pH. There may also be some correlation there with the amount of biomass. If there's too much or too little, there just may not be enough metabolism there in order to keep things going. So then we're moving on and looking at a few other inhibitors. So instead of just inhibiting the proton motive force, we wanted to inhibit the specific complexes. And so the first of the reverse electron transfer inhibitors that we looked at was Antimycin A. And what this should do is inhibit the BC1 complex. And what we see here is a much smaller drop in the current. So this here, it's 3 to 8%. And one of the problems here is that when I added it, a lot of it precipitated, so that we may not have actually been adding enough or getting enough of this inhibitor to the cells in order to block this. And we see the same thing if we look at this other inhibitor, rotenone, which inhibits, it's essentially the complex 1 in the bacteria. And again, we see this 3 to 8% drop in current, but we also had a lot of precipitation. So it's hard to really know for sure, but the thing that does interest me about this a lot is that if you add the two of them together, so I added rotenone and then added antimycin, and I saw around 5% drop when I added rotenone, and then I saw another 5% drop when I added antimycin. And I saw the same thing if I added antimycin first and then rotenone. So it looks like there is something happening there, but it's not enough to completely get rid of it. So still need to work on that a little bit more. But then if we add cyanide, this completely kills the current. And this makes a lot of sense because we think that hemes are very critical to everything that occurs. So they're extracellular heme or outer membrane hemes that are required to get electrons across the membrane. There are periplasmic hemes that are required to get it from the outer to the inner membrane. And then there are hemes and several of the enzymes that are involved in the membrane-bound electron transport chain. And so when we add cyanide, all of these hemes get poisoned and we see this immediate drop in current. And the reason it doesn't go to zero completely is that there's actually a slight offset here. So if before I added any cells to this reactor at all, the current was slightly positive just because of the way that there was some variability in the reference electrodes that I was using. So this amount of current that's generated after we add the heme is identical to the amount of current that was generated before I added any cells to the reactor at all. So from this, using inhibitors, we've been able to get this real-time data on the electron transfer processes in this community. And the CCCP data gives me a lot of confidence that the model that we're using is actually correct, because we see this drop in current that is identical to what we would have expected. I need to work a little bit more on the complex 1 and the BC1 complex inhibitors, because the precipitation just isn't allowing us to see what we want to see there. And this is work that I'm hoping to be continuing on in the next several months here, but it's been kind of pushed to the background for a while. So from here, some of the other things that we're interested in working with still and have in progress is we've got a lot more sequencing. We're doing transcriptomics on isolates. We're hoping to do more transcriptomics on other conditions within the biofilm, trying to grow trying to isolate Tenderia still. We've been working on that for years, and it just hasn't been something we've been able to get in pure culture. We're also working very hard on developing one of the isolates from the community that is called Marinobacter CP1 as a chassis for synthetic biology. And we've made a lot of progress along that front. And then we're testing some of these components, or we would like to test some of the components we found with the metatranscriptomics and metagenomics, trying to put them into other organisms and see if we can recapitulate this electron transfer there. And so with that, I'd like to thank the people that I've been working with, particularly Sarah Glavin, who was the PI for a lot of this work and was the one who hired me at NRL to begin with. And then Baochuan Lin was also a co-PI for the first several years I was at NRL. And then Kirsty Coombs did a lot of the reactors for the inhibitor studies. Matt Yates did a lot of the electrochemistry. With that, I would be happy to answer any questions that you have.
Thank you, Dr. Eddy, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, why do you think you can't get Tenderia into a pure culture?
So one of the things that a lot of iron oxidizing bacteria have a very high sensitivity to is organic carbon. And one of the ways that we normally isolate bacteria is using agarose plates, or for some iron oxidizing bacteria, they'll use iron gradient tubes. And when you autoclave to sterilize the agar, sometimes you can actually break it up and make some smaller sugars that then will end up inhibiting some autotrophic metabolism. And so that's one possibility. Another possibility is that there's just something that one of the other organisms within the community is doing that then is helping them out. So it's either making some sort of vitamin or cofactor that they need. Another possibility is it's detoxifying something. So some iron oxidation metabolism is making some reactive nitrogen species, like nitric oxide or nitrite. And then there are bacteria that can then use those and detoxify those. So that's another possibility.
Thank you. Is it normal to have more than one rubisco in the same organism?
Yeah, there are a lot of bacteria that have multiple types of rubisco, and they just each have their own utility. So like we see with Tenderia, the one is more highly effective when there's a lower concentration of oxygen and a higher concentration of CO2, and the other is better when there's less. So they just each have their own uses. I think there's one organism that actually has 4 different rubiscos.
Interesting. Thank you. So it looks like we have time for one more question. What other inhibitors of reverse electron transfer are out there that you could try?
So there are other inhibitors of the complex 1, particularly. So some of the things that we'd like to try are using azide, which inhibits hemes that it's a lot like cyanide, where it inhibits hemes and it binds to them irreversibly. And then there are also ways that we can actually block the forward using zinc with the reverse electron transfer. I actually can't remember. Yeah, there are a couple more out there. I'll try and get back to that one.
Very good. Maybe people will even email you about that. Yeah. I would like to once again thank Dr. Eddy for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 13th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.